My name is Dr. John Meinert. I'm an assistant professor of theology here at FranU, and I'm very excited today to welcome Father Nicanor Austriaco, who is the author of many, many studies, over 50, co-PI, theology author, scholarship, and an author of a textbook I use on bioethics in class. And I'm very excited to hear him talk about CRISPR gene editing technology. This is something about which we need to be thinking, especially as a healthcare institution. So without further ado, Father Nicanor.
Thank you so much. So, several of you were at my talk last night. It's going to be very different, because then we were just talking about the theological implications of evolutionary theory. Today we're going to be moving from what is called the dogmatic or doctrinal account of theology to moral theology. How should we live a good life? And how should we live a good life in light of human genome editing? So what I'm going to do is, the talk is going to be divided into 3 parts. I'm going to begin with the science. I'm not sure where everyone is with regards to the science, so I'm just going to start from scratch. Talking about how we can edit genomes today. Then we're going to talk about a couple of distinctions. These are contrasts that will allow you to think through the issues, and we'll move to the ethics. So we're going to begin with the science, and this is a slide I used yesterday, but in this particular context, I want to just talk about what DNA looks like and how you can imagine the editing that happens. So we have 46 chromosomes found in the nucleus. The nucleus is the central command component of the cell. We inherit 23 from mom and 23 from dad. We also have a small genome of 37 genes that are in a small circular piece of DNA that is inherited through the mitochondria, which is the powerhouse of the cell. And if you take one of these chromosomes out, what you will see is that the chromosome is basically the iconic double helix. And you have these base pairs, and each base pair is composed of a pair of molecules that are able to interact at a chemical level. And so we basically can depict DNA as text. So this is the sequence of the gene that my students and I have been studying for the past 7 years, BAX inhibitor, which is overexpressed in prostate cancer and several other cancers. And we now know it encodes a pH-sensitive calcium leak in the endoplasmic reticulum. And my students and I are just beginning a small molecule inhibitor screen looking for inhibitors that will prevent this protein from functioning. Nonetheless, the purpose of this slide is to show you that DNA can be depicted as text, and so we can edit DNA in the same way that we can edit text. So this is CRISPR. It's a molecular editing machine made up of 2 components. There is an RNA component called the guide RNA, and what this RNA does is it targets the cutting machine directly to a very precise spot. And Cas9 is the protein component, and that's actually the cutter. And so you can imagine a pair of scissors, and the pair of scissors is attached to a guided missile, and the guided missile directs the pair of scissors to a particular place in DNA, and it makes a cut. And what we can do is that we can do a whole bunch of different things with a simple machine, but we're going to focus primarily on the editing part. When biologists talk about the editing, what we do is we point out that all that the CRISPR-Cas9 machine does is it makes a cut. That's all it does. It makes a single cut in the double-stranded DNA helix. It's what happens after the cut is made that is important. If the cut, the break, is simply repaired, and it can be repaired, then what will happen is that some information in that gene is going to be lost or destroyed. And what happens here is you can basically get rid of a gene of interest. The alternative way is that in the process of making the cut, you provide the cell with another piece of DNA that will now be used to correct the cut. And so you can change the sequence, the bases. Say, for example, a G to an A or a T to a C. You can then change it. So on this side, you destroy. This side, you revise, you edit, you alter. And we can do a lot with this today because of its incredible precision. You can get down. If you think about the genome, it's 3 billion letters organized into 23 volumes. So think of the genome as an encyclopedia. With this machine, you can go in to page 422, column 2, paragraph 4, the third word in, and change a single letter of that entire encyclopedia so it fits, so it conforms to what you want it to do. That's how precise this is. Now, where did we get this machine? This machine evolved in nature, and it evolved primarily as a bacterial cell's immune system. In order to understand how this works, so this red line is a cell, it's a bacterial cell, and that's a virus, and the virus injects its DNA, and this virus is going to want to take over the host cell's DNA in order to make more copies of itself. If the cell survives the first infection, what it will do is it will take the DNA that was introduced by the virus and store it in the CRISPR locus. That's a position on its own DNA where it stores information of viruses that have previously infected it. So upon a second infection, what the cell is able to do is it uses the CRISPR machine and the stored information that it stored from the first infection to cut and destroy the invading viral DNA. And so that's why this machine cuts, because it was intended to cut the invading DNA. But what we've done is we've now just taken the machine out of the cell, out of its original biological context, and used it as a tool. We are using it as a tool to change the DNA, not just of bacterial cells, but any cell that you can find around you. So the original papers were published in 2012. In fact, I was wondering whether or not the Nobel Prize would be given. So it is expected that the Nobel Prize will be given. It was thought maybe this year, which happened a couple of days ago, or maybe next year. And I think what they're doing is they're waiting for the resolution of the patent battles. Honestly, I think what's going to happen is that there's a patent war that's happening between MIT and the University of California, San Francisco. No, it's Berkeley. And so what's happening is you've got this, they're duking it out in the courts. The appeals court recently gave the patent to MIT, the Broad, and I think it's going to be appealed. And I think that you're not going to see a Nobel Prize awarded until the legal battles are completely resolved. Because I don't think the Nobel Committee is going to want to insert itself right in the middle of a legal battle. But the expectation is that Jennifer Doudna, Charpentier, Emmanuel Charpentier, at least those two, if not a third, are going to receive the Nobel Prize for this discovery because of a seminal paper that they published in 2012 where they were able to show that this bacterial defense system could be used to edit human genes. So since then, we've been able to edit mice, fish, monkeys, and plants, rice, and as you will see in a second, we've been able to edit human genomes as well. So to give you a sense for how you can use this to edit disease, so this is a mouse model, and it's got, this is its liver, and the mouse is carrying a mutation that makes it unable to metabolize tyrosine, which is one of the amino acids that is important for protein metabolism. And so this is the mutation that gives rise to human hereditary tyrosinemia in patients. And what scientists have been able to do is they've been able to genetically alter some percentage of the livers of the mice, so that the mouse is able to so it repairs the liver, so the liver is now able to metabolize tyrosine once again. A very controversial proposal using CRISPR is the whole notion of gene drives. Here, you are able to edit a single animal, and you're able to edit in this case, the liver of a single animal. The whole notion of gene drives is to edit an entire population of animals. And so the idea here is to use CRISPR in order to eradicate one of the 3,500 species of mosquito. The species of mosquito that is primarily responsible for the spread of Zika virus, malaria, this is Aedes aegypti. Now, the way this works usually is that if you've got a mutant mosquito mating a normal wild-type mosquito, there's a 50-50 chance that the offspring of the mosquito will inherit the gene. But using gene drive technology, what you do is that when the mosquito inherits the gene of choice, CRISPR will convert the other version of the gene into the version that you want. And so what ends up happening is over several generations, you can actually genetically engineer every single individual in the mosquito population. And the way they're doing this is they would like to introduce a mutation that alters the reproductive capacity of the mosquito. So here you basically make all the mosquitoes sterile, and you wipe out one species of mosquito forever. And so what ends up happening is you get rid of the insect vector that spreads Zika and malaria. Now, you can imagine that this is incredibly controversial. Now, my students ask, have we ever done this? In a sense, we have, because we've already wiped out smallpox. So we've wiped out one particular virus, and the hope is to get rid of polio. And so the question is, how is that different from eradicating a single species of the 3,500 species of mosquitoes? The idea is that you're not getting rid of all mosquitoes, you're getting rid of the one kind of mosquito that is a disease vector. And in the process, get rid of malaria, get rid of Zika. So you can do a lot there. But the ecological impacts of that are unknown. Okay? So this is part of the ethical debate that we're going through. Now, with regards to human genomes, you can do it in two ways. The more common way that is being explored today is the second way. This is called ex vivo editing. And what happens here is you take stem cells out of the patient, you genetically engineer, using CRISPR, those cells outside the patient, and then you put the cells you want back into the patient. So the editing happens outside. In vivo editing is the editing happens inside your body. And the goal here is you have to take a virus, and the virus carries with it the CRISPR editing machine. You find a way to target that virus to a particular tissue in the body of the patient. It goes there, it infects cells, it edits those cells. So you have in vivo editing and ex vivo editing. A lot of work is going into ex vivo editing at the moment because this is considered safer than in vivo editing, because here you have more control. You can actually decide, you can screen out non-edited cells or cells that were edited inappropriately in the hopes of reintroducing into the patient only those cells that were edited absolutely correctly. One of the things that's being done, for example, is to try to correct sickle cell disease. For those of you in the healthcare professions, you may be aware that what sickle cell disease is a single genetic mutation where a T, where the normal A has been replaced with T, leading to the sickling of the cells because of a defect in hemoglobin. And the goal here is to use CRISPR-Cas to alter the single letter from T back to the normal A to restore healthy blood cells. You would do this in stem cells found in bone marrow, and then you would replace the bone marrow of the patient with this edited version in order to cure the patient of the disease. Another area that people are working on is to correct inherited genetic retinal diseases. So these are diseases that inevitably lead to blindness in patients. And what you're seeing here is that you're able to, what they do is they will inject the CRISPR editing machine directly into the eye, and directly into the retina in the hope that the machine will correct some percentage of the retinal cells. So it turns out in order for you not to go blind, you only need about 10% of the cells in your retina. And so if you've got a machine that's able to correct 12%, you would be able to significantly improve the quality of life of patients who are imminently going blind, and you're able to keep them from doing so. And so the idea here is to inject the DNA or the machine directly into the retina. Now, I mentioned to you, this was in 2017, this is in the fall of 2017. There was gene editing in human embryos in the United States. This is in Shukrat Matalikov's lab in Oregon. And I'm not going to go into the details, but what they were able to do is they were able to introduce CRISPR-Cas into a human embryo and correct a genetic defect associated with in a disease that has been linked to congenital to heart failure, heart defects. And they tried two different approaches. You inject the CRISPR-Cas after fertilization versus you inject the CRISPR machine simultaneously with the sperm, and it changes the way that the editing occurs in the developing human embryo. So now let's move to distinctions, just things that you need to keep in mind before we jump into the ethics. So we already talked about ex vivo versus in vivo, and you can see that a lot of people want to deal with the ex vivo before the in vivo. They're concerned that once you introduce the genetic machine into the patient's body, you don't have control of that machine, and it could edit more than you really wanted to edit. The second distinction that's really important is somatic versus germline. And somatic cells is to change and edit the genomes of any cell in the body other than the reproductive cells. So in this case, you can change the lung cells, the muscle cells, the liver cells. But in germline gene therapy, you're actually changing the cells that give rise to the next generation. So this would be gene therapy that will be inherited by the patient's children. And the question people here are asking is, do we have the right to alter the genes of future generations? Should we limit ourselves primarily to patients who are living here and now? Do we have the right to alter the genes of tomorrow's people? There's another distinction that's often made, therapy versus enhancement. People assume that if it's therapy, it's good. If it's enhancement, it's not good. And this distinction is actually used by the Catholic Church when talking about the ethics of any sort of genetic engineering. And so this is the document Dignitas Personae, published by the Vatican, the Congregation for the Doctrine of the Faith. And so the idea here is that therapeutic purposes, procedures used on somatic cells for strictly therapeutic purposes are good. The assumption is that if they're for enhancement, they're not good. And the idea here is that you are going to restore the person to the normal genetic configuration of the patient. That's what therapy is, to restore the norm. Anything outside the norm is considered enhancement. Now, and so this is now dealing with enhancement, improving and strengthening the gene pool, and this is considered outside the bounds of moral procedures. Now, I'm just going to put this slide on. Just, people, they ask the question, like, if you ask moms and dads, if you were allowed to genetically engineer your kid, what would you do? So, a lot of people, so here on the left side, you've got parents who believe that gene editing is good. They'll say intelligence, 28%, health, 27%, creativity, attractiveness, and weight. On the right, these are parents who are suspicious of gene therapy, and yet they will say, again, health, intelligence, weight, creativity, and kindness. And this is particularly, especially with intelligence, there is no single gene that regulates intelligence. It's really a constellation of factors that come into play. So it's unlikely that you're going to be able, in one genetic change, to radically improve the IQ of an individual. But people don't know this, so they just assume, I just want to make my kid Einstein smart. But here's the question, is the distinction between therapy and enhancement a robust one? I'm going to propose to you that it's not, and that we already violate this distinction today. Because I asked this question, what should the target LDL levels be for patients at risk for cardiovascular disease? So for those who are not clued in, LDL is bad cholesterol, HDL is good cholesterol, and as you get older, your LDL levels go up, and everyone wants to drop it down, and statins is it. And we prescribe statins in order to lower LDL. But what should we target LDL levels to be? So our average LDL level amongst US adults, 119 milligrams per deciliter. And the normal range is 90 to 130 milligrams per deciliter. And yet, when you talk about the recommended standard of care amongst cardiologists, they're just going to say, lower the better. Because there's some really good data that suggests that the lower the LDL, your risk of cardiovascular incidents go back, adverse cardiovascular incidents go down. So if you look at hunter-gatherer tribes, 70 milligrams per deciliter. If you look at non-human primates like gorillas, 30 to 50 milligrams per deciliter. And there's actually a healthy human patient with a mutation in PCSK9. He is going 15 milligrams per deciliter. So what should the target be? What should the target be? Now, if you believe that therapy is restoring to the normal, then we should target 90 milligrams, even 120 milligrams. But we should target 70 to 130. But that's actually not we already don't do that because we know that the lower it is, the better. And so what cardiologists do is they just go lower and lower and lower. And so the standard of care right now is we should lower LDL levels even beyond the species norm to preserve and protect human health. So what you're seeing here is we're already enhancing patients, not genetically, but pharmacologically using statins. No one's complaining. There are no ethicists out there who are saying this is bad because we're trying to make patients better than normal, better than well. So I don't think there's a difference. I think there are therapeutic enhancements. These are enhancements that promote the human good of health. and this is one of them. You're looking at statin use, and the idea here is that we're going to make you better than what the norm should be, and there are people who are saying we should get you down to 15, because we can see there's a patient with 15, and he's completely and totally healthy. So the distinction I'm proposing is that it's not enhancement versus therapy, it's therapy versus non-therapy. And when people ask, what is non-therapy? So if therapy is for advancing the good of human health, when people talk about non-therapy, I say it's about opportunity. Parents want to make their kid 6'5", because they've heard somewhere that tall people tend to be more successful, because human beings tend to respect, admire, etc., taller people. And so they want to enhance the personal and or social opportunities of individuals. That's non-therapy. Now, if this is the case, therapeutic enhancements like statin use are good, then can we not talk about genetic immunization? So today you get immunized against all sorts of things. The question here is, can you be immunized genetically against HIV/AIDS? So some of you may or may not know, there's a subset of people in the population that are immune to AIDS. They're called elite controllers. They carry a mutation, CCR5 delta 32. So it's a deletion of a certain portion of the gene, in the gene CCR5. They can become HIV positive, but they will never progress the disease. And so one of the things that's really striking is, can we use this mutation to immunize patients against HIV/AIDS? Now there is a very interesting case of an AIDS patient who developed leukemia, and people were so distressed that he was struggling with 2 life-threatening conditions. But this is in Germany, but his doctor was incredibly smart, because he had to undergo a bone marrow transplant, and the doctor said, you know what, I'm going to find someone who matches you, but who's also CCR5 delta 32. So what they did is he received a bone marrow transplant with bone marrow cells with this mutation, and not only was he cured of the leukemia, but he was also cured of the HIV/AIDS, because now what happens, he stopped taking the drugs, but because of his rebooted immune system carrying this mutation, he was an elite controller. And so there are people who are talking about doing something like this to immunize everyone against HIV/AIDS. It's a very interesting proposal. Now, you may or may not know that it's very hard for elephants to get cancer. And one of the reasons is they have 20 copies of P53, while you and I only have 1. And so they have so many, and P53 is it's called the guardian of the genome, because in order for you to get cancer, you have to get rid of it. So if you have 1 copy, or 2 copies, 1 from each parent, you need 2 hits in order to get rid of this gene and develop cancer. If you're an elephant, you have 20 copies, it's much more difficult to get cancer, because then you've got to get 20 bullseyes in the exact same cell. Not easy to do. So the question now is, can we introduce extra copies of P53 into human beings in order to immunize them and prevent them from ever getting cancer? So these are the kind of questions that people are speaking about with regards to CRISPR. Because with CRISPR, in theory, you would be able to do this. You would be able to introduce extra copies of genes into the genomes of individuals in order to keep them from ever getting cancer. Now let's move to ethics, and I'm going to speak here primarily from the perspective of a Catholic bioethicist, because there are 2 paradigms of ethics in healthcare in this country. So you have the Catholic, the faith-based approach, and then you've got the secular approach, and I'm going to come to this as a Catholic Christian, but I'm going to put it into conversation with the secular account of ethics so you can see the similarities and differences. So there's a lot of conversation going on right now about CRISPR and the ethics of CRISPR because of this DNA revolution. Now, what would a faith-based approach look like? So just, coming to this question, what is primarily different between a faith-based approach, and we're dealing here not just with Catholic bioethics, but often with Jewish bioethics, with Muslim bioethics, a faith-based approach versus a secular approach. How would they be similar? How would they be different? And it comes down to this key concept called human dignity. And it's interesting because in the very first sentence of this document, this bioethics document from the Vatican, the dignity of a person must be recognized in every human being from conception to natural death, which is why the title of the document is Dignitas Personae, the dignity of the person. Now, a lot of people assume that the human dignity is a theological, a faith-based account of the human person, but if you look at the first sentence of the German federal constitution, the Grundgesetz, the very first sentence, the opening line of the German federal government's constitution is, human dignity shall be inviolable. Now, there are two reasons for this. The Constitution was formulated and written, the basic rule, right after World War II, and you can imagine that the German state was responding to the atrocities and gross violations of human dignity, which is why they put this up in front. And it's also clear historically that the German state, when they were formulating, were heavily influenced by Catholic social teaching with regards to understanding what constitutes a state, what constitutes the human good, what is the relationship between the government and its citizens. So it's not simply just a Catholic proposal. It's a proposal that has been present, that is now present in the Western philosophical tradition. However, it is disputed, which is why I end up going to Germany several times a year in order to speak about human dignity. Because this is the dispute. What is it? What is human dignity? And the way I deal with this when I go to class is I go, it's an answer to this question, how much are you worth? And if I ask you, how much are you worth, what kind of answers will you give me? How much are you worth? Yeah, especially if I ask your mom. Because I ask how much you go, I don't know. I go, what would your mother say? Priceless. And I'll say, that is an account of human dignity. Right? And it's not just a theological, even, what Kant said. So Immanuel Kant, great German philosopher, he would say, where things have price, persons have dignity. And what he was trying to indicate there is, we are beyond price, because there's something incredibly valuable, incredibly priceless about it. So the way I explain this is I say, how much is an iPhone worth? And I have to specify 32G, because those are all the range, right? And if you look, there are two numbers associated with that. So the teardown cost, which is, and you can Google this teardown cost, and I have not even bothered to look at the iPhone XS Max. That's the new one. The teardown cost is where people buy the thing, engineers buy the thing, then they disassemble it, they open it up, and they calculate how much it costs for basically all the parts of that thing. And so the teardown cost, the intrinsic value of the phone, is $219.80. The manufacturer suggested retail price $614. Now there's a huge markup in Apple products. Okay? So notice the things about this. This price doesn't change. This price is the price of that phone once that phone comes into being, and it will cease to be the price of the phone only when that phone falls apart. This price, on the other hand, this goes up and down. It's the Friday after Thanksgiving, this price is probably going to go down. There's a run on iPhones, this price is going to go up. So this price is sensitive to supply and demand. It's sensitive to the social environment that you find yourself in. This one is not. And this is why we call this intrinsic and this extrinsic. And the same thing, many faith-based, including the Catholic tradition, will say you have both intrinsic and extrinsic worth. So, intrinsic dignity, your mom is correct. You are priceless. But you have extrinsic dignity too. Usually today, that is pegged to your salary. So, if you have a higher salary, then you are worth more to society. Now here's the issue. The secular account of bioethics has no place for this. The only thing you've got is this. And that leads to a problem, because you can gain or lose dignity depending upon the circumstances of your life. You have Alzheimer's, end state, your salary is practically zilch. And your net worth, well, if your treatment has basically emptied you of your savings, your net worth is zero. So what ends up happening is a lot of people end up saying, I've lost my dignity at the end of my life. So, but you see, that's because they're talking about the extrinsic dignity. We're also talking about intrinsic. Same thing with celebrity or high office, right? So we talk about, well, Lady Gaga, celebrity there. She has higher extrinsic dignity than most of us in this room. But here's the thing. Should extrinsic dignity drive bioethics? And the question I ask here is the same thing, you know. The President of the United States comes in to the hospital, and the mother of 3 kids comes in. They both need a new heart. Should his being president, that's extrinsic dignity, give him an advantage over the mom of 2 kids? Now, in practice, we say no. Because in practice, even though we live in a post-Christian society, there is still an account of intrinsic dignity that we don't want to get rid of. And so we say that the heart should go to the sickest patient, regardless of who that patient is. You see, that account is recognizing the intrinsic dignity of the patient. If it was mere extrinsic, you'd say the more important, the more valuable, the richer you are, you should get that heart. So you see, in bioethics, there's this tension. Now, if intrinsic dignity is the worth that you have simply because you are human, right? Then it will be there as soon as you become human, which is why it has to begin at fertilizing conception. Your intrinsic worth, regardless of what you can do, if it's attached to your being human, this is why the Christian tradition affirms that the value of the human being begins at conception, because it's like the phone. The intrinsic value of that phone can be attributed to that phone as soon as the phone is put together. It doesn't have to go on sale. No one has to turn it on. It simply is a phone. It has an intrinsic value. If it's an iPhone 7, $219.60. And it will cease having that value only when it falls apart. And for us, we call that death. So you see, this is the philosophical basis. A lot of people will say, well, the Catholic Church's position against abortion is theological. And what I'm trying to show you is it's theological and philosophical. There's different ways of looking at it. In this case, notice, intrinsic dignity is not earned or bestowed. It is only acknowledged and recognized, which is why it's so important to affirm this when you're dealing with governments that are trying to diminish human dignity. And we're hearkening back to the Nazi government during the tragedies of World War II. What you see there is, this is why the German state wants to affirm an intrinsic account of dignity. Because if it's intrinsic, as soon as you are, it doesn't matter who you are, how much money you make, what religious background you come from. Because you are human, you are valuable. That's the intrinsic. Notice, it's also absolute. You can't be half an iPhone or a third of an iPhone. It's either you're an iPhone or you're not. And the same thing is true about persons. You're either a person or you're not. You can't be half a person, a third of a person, a quarter of a person, which is what a lot of people are talking about when they say, well, you become more and more of a person as you develop in your mother's uterus. But that, you see, is an extrinsic account. It doesn't deal with intrinsic. Same thing, there is no such thing as a partial intrinsic dignity. You either have it or you don't. Now what's striking is that for Christians, intrinsic dignity is grounded upon a theological claim made in the image and likeness of God. But it's not just merely theological, because you can also explain why we are we are priceless because of the kind of things we can do. So yesterday, in my talk in evolution, we talked about how human beings are rational. We can abstract. We can do things that animals can't do. Now, I have no time to jump into how this is worked out, but once you recognize that you and I and we alone can talk about abstract realities, this necessarily points to us having an immaterial soul. And once you have an immaterial souls are like Louis Vuitton. They're just incredibly valuable in themselves. And so what actually happens is our value can be attributed to a theological claim. We are made in the image and likeness of God. It can be attributed to a philosophical claim because there's part of us that is eternal. And once, not immortal, and once we are immortal, being immortal is priceless as compared to being mortal. But that's a whole different philosophical argument. So, for ethics, in the Christian, Catholic, and I've talked to Jewish and Muslim, very similar, you have to protect both the intrinsic and extrinsic human dignity. It's not just extrinsic dignity, we have to deal with intrinsic dignity as well. So, and human actions that fail to do this would be ruled out as immoral. So now let's just talk about the secular approach. I'm proposing to you that the primary disagreement between a Christian bioethics and a secular bioethics has to deal with this dignity. So this is a very famous article by Ruth Macklin, a secular bioethicist in New York. The title, "Dignity is a Useless Concept." And what she wants to argue is that you have to replace dignity with respect for persons or their autonomy. So autonomy is an autonomy-based ethics rather than dignity-based. And autonomy means you get to choose whatever you want to do unless it hurts someone else. And this is kind of like the rule that everyone has today in our society. You're free, you're independent, you're autonomous, you get to do anything you like as long as it doesn't hurt anyone else. She's not the only one. Steven Pinker, a professor at Harvard, the stupidity of dignity. Again, the claim here is it's not dignity, it's autonomy. Respect for personal autonomy. But then you see, we have to ask this question. Why is autonomy worthy of respect? Why should I respect autonomy? And I'm going to speak now as a biologist. Autonomy is the ability to choose. Now, it's interesting. In order for you to choose, you need to have the ability to think and the ability to will. So this is to desire. Are evolutionary adaptations that are not more valuable than other adaptations. And I give you this example. If tomorrow you found out that you were going to be in the middle of the Siberian tundra, would you choose to be a human being, naked, cold, but autonomous? Or would you choose to be a polar bear? Most of my students say, of course I want to be a polar bear. And so one of the things you discover is that being a polar bear is more valuable in the Siberian tundra than it is to be an autonomous human being. Now in Baton Rouge, you do not want to be a polar bear, because if you were a polar bear, you would soon be a dead polar bear. So being an autonomous human animal is advantageous in a city, but it is not advantageous in the tundra, and vice versa. So what happens is there's no reason to believe that being an autonomous individual is intrinsically more valuable than flying or swimming or eating fish, because it all depends upon the particular ecological niche you find yourself in. See, this is Darwinian through the core. But if that's the case, then why are you and I special? Right? This is the thing. I grew up in Bangkok, Thailand, and one of the things that's striking when you grow up in a society where the gospel has not really penetrated the culture is that you have, this is a long time ago, like a girlfriend, long before I was a priest. She was Buddhist, and what's really striking is if you believe that you could become a mosquito in the next life, if reincarnation is something that's at the heart of your worldview, and that you could become a mosquito, then this mosquito could be your great-great-grandmother. And if this is the case, then that mosquito is worth as much as you are, because you could be a mosquito. And so what ends up happening is that there is no way to account for what we take for granted is the exceptionalism of the human animal. And we just take that for granted. If I can kill a mosquito, and I've done that many, many times in my life growing up, and if a mosquito is of equal worth to you, then why can't I kill you? Do you understand? Now, because most of you come from a Christian account, even if it's post-Christian, you simply assume you're worth more. But biologically speaking, there is no reason to think that we are worth more than mosquitoes. First thing. Second thing, as you'll see in a moment, it's not quite clear to me why we are all equal. The American, we are all equal. Why are we equal? Come up with one standard. The very nature of the university is to show you you're not equal. We have grades. And grades, by nature, tell you you're different. And if you look up any single measure, there are people who have more of that measure and less. If you're talking about pretty, they'll be really pretty, not so pretty. You're talking about smart, you get really smart. You're talking about weight, unfortunately, a lot at the top, not so at the bottom. There is no one thing that we all have that is the ground for what we affirm with the conviction of the American soul that we're all equal, except for intrinsic dignity. You see? So this is the thing. So dignity has a lot of explanatory power. It explains why we're equal, regardless of genetic, cognitive, social difference. There are going to be all these differences, usually on a normal bell curve, but we never say that you are more or less. Now, we have a debate in my theology class because I point out that, so we had a debate just the other day before I flew out here on Wednesday. Do parents love all their children equally? Now, as a priest, talking to many, many parents, I know they will whisper, "I have a favorite." And I tell my students that they're all freaked out. They're like, "What do you mean?" I'm like, "Yeah, your parents are favorite." And then one of my students goes up and goes, "I knew it. I knew it my whole life." And he goes, "I am not the favorite." Now, it's really striking because you see, because each of us is different, there are certain things that you like about this kid that you don't necessarily like in that kid, right? And so my students were like, "Well, how do you know which is the favorite?" And I go, "First of all, your parents are going to work really hard never to tell you which one is the favorite. Never, never, never." But think of it this way. You have a 24-hour drive to Florida. which one does your mom, who does your mom want to go in, your car or his car?
Yeah.
You see what I mean? They go, and they just she's gonna hang out with me. And then one of my students goes, that's because I'm the only girl. She likes me, I'm the favorite. But you see, it's really, really striking because when you realize that there is nothing other than an intrinsic dignity that grounds our equality. And it's not like parents love intrinsic dignity. They don't say, I love your intrinsic dignity, I love your intrinsic dignity. They're all exactly the same. What it makes you realize is that they're different. And this is a normal part. And I tell them, your parents have favorites, but you are still equally their kids in their eyes, you see? And this is very striking them. Same thing too. Why are you worth more than a kangaroo? Now, I have students who are not sure that they are worth more than a kangaroo. We live in a day and age where no one's sure anymore. You know, there are chimpanzees are suing in the courts for rights because we're not really sure anymore. I mean, there was a very interesting situation a couple of years ago because a woman in England wanted to marry a dolphin. She had fallen in love with a dolphin in the aquarium in Jerusalem, and she said that she should be able to marry anyone she wants. So she goes, this dolphin, I love the dolphin, he obviously loves me because every time I come, you know, go visit him in Jerusalem, the dolphin comes, and people were asking, well, you know, he's also got other dolphin mates, she's totally okay with that, she's not jealous. As long as she's the only one, the only human mate. Now you see, this is really interesting because my students struggle to explain why, and they'll say, especially with dolphins, because they're like, dolphins are smart, dolphins can communicate, why can't you marry a dolphin? There's a lot of people who are not sure anymore, okay? Because the idea is that, and the answer is, she's not hurting anybody, and she should be given the freedom to choose. So if she chooses to marry a dolphin, now the one thing that I got back was, well, we don't know if the dolphin wants to marry her. So now what's happened is students are saying, we have to look at the autonomy of the dolphin. You see, because the whole thing is driven by autonomy. It's about choice. So she shouldn't be able to marry the dolphin because the dolphin, you can see part of the confusion in our culture because we are denying the existence of intrinsic dignity. So, for faith-based bioethics, dignity is the ground for autonomy. You need to have dignity first before you have autonomy. The only reason why we recognize your autonomy and not the dolphins is because you have intrinsic dignity, the dolphin does not. And then once this is the case, dignity can limit and constrain autonomy. You cannot do anything that will attack human dignity, even if it doesn't hurt, quote, anyone else. You see? And this is where people disagree in our day and age. So now, we'll talk at the very end about CRISPR, how do you put this all together in a couple of slides. Human genome editing with CRISPR to advance the dignity of the human person. So, there are 4 different categories of concerns associated with dignity concerns with human genome editing. First of all, you have to make sure that persons are safe. Second is your concern about commodification. So let's talk about safety first. We're worried about off-target mutations. So the CRISPR machine comes in and does a cut. Will it do cuts anywhere else? What is so striking is I was speaking to scientists from one of the CRISPR companies in Cambridge, and they asked a simple question. How many cuts are there normally in any human cell? It turns out there are thousands. And so we seem to think that the DNA is totally perfect and the machine comes in and cuts. It turns out there are cuts everywhere, which makes sense. You have a 7-foot piece of DNA wrapped really tightly in a cell. It's going to get damaged by mechanical, physical, chemical forces. So there's always cuts. And so the question is, will CRISPR enhance the number of cuts? Chimeric tissues is a concern. We're talking here about if you have a liver, what percentage of the liver will be genetically edited? Is it 10%, 20%, 30%? And chimeras is important because you want to make sure you edit enough of the organ so that the function that you are trying to seek out will be restored. Now, commodification of persons, commodification is to treat a person as an object. Now, this is, remember, things have price, persons have dignity. So anything that puts a price on the human person will be considered ruled out. Which is another reason why, in addition to the biological, so when I talk to my students about abortion, I say one of the reasons that abortion is problematic is because it puts a price on the baby. And they're like, what's the price? The mother's choice. That she decides how much value that baby has. If she says it's valuable and she keeps the child, then what ends up happening is that if she is killed in some way, the individual kills her is guilty of 2 homicides. But if she chooses not to give value to that person, then what ends up happening is that the physician who helps her to terminate the pregnancy is not considered someone who is guilty of homicide. So you see what happens is the price of this person, in our culture, the price of the human being prior to birth is attributed directly to the choice of the woman. And I'm a Project Rachel priest. I'm a priest who has the privilege of accompanying women and men who struggle with abortion regret. And one of the things I've come to see is that the choice that a woman makes is tragic, incredibly difficult, incredibly painful. Now, for some women, it is not a difficult choice at all. But for about 1/3 of the women, it comes back to haunt them. And so this is where you can see, and I will say, part of the reason why abortion is morally problematic, for many reasons, but one of them is women themselves are damaged. Women themselves are hurt. Men too. So I've had conversations with men who have felt emasculated and incredibly diminished because they were unable to, in their minds, protect the lives of their kids. Same thing, I don't have a chance here to talk about artificial reproduction, but one of the reasons why the Catholic Church finds some of the procedures associated with assisted reproduction problematic is because it objectifies the human being. And here, the person is not made in the image and likeness of God, but in the image and likeness of another person. Now, with regards to the marginalizations of persons, this is particularly important at a Franciscan university. The difficulty is that you're going to exacerbate social divisions and inequality. So here, the rich are going to have access to CRISPR, the poor will not. So the rich will become more beautiful, the poor will not. And so we'll exacerbate the struggles between the two. And then you're going to have the eugenic temptation. So I was giving this talk last week in Chicago, and I met with a disability advocate who was deeply troubled because she is concerned that attempts to eradicate disability will diminish who she is as a living human being. And she came to speak to me primarily about the selective abortion associated with Down syndrome. You may or may not know that in Iceland there's only one child with Down syndrome because every other child, the government has begun a process of eradicating Down syndrome from the population using selective abortion. What happens is they miss this one. And she's the only one in the entire country now. And this is going on all over France is, I think, 75%. They want to get it 100%. So the idea here, and then this individual who belongs, the person I was speaking to who belongs to the disability community, is deeply troubled that this eugenic mentality, this mentality of, we're going to get rid of everyone who's imperfect, is going to undermine her place, her role in the society that we live in. And then we have to make sure there's a preferential option for the poor and the vulnerable. And so when I talk to companies working with CRISPR, I say, how are you going to make sure that your technology is going to be available in Africa for people who need it there, especially for therapeutic reasons? So these are all the things. So finally, what I'm going to do dealing with somatic cell, I have written on this. So basically, somatic cell modifications for therapeutic reasons, I think can be morally justified. I even think it can be more morally justified for germline modifications, because if we can cure you, we might as well prevent you from getting sick. As long as this can be done in a way that does not objectify the human person. But anything therapeutic, non-therapeutic, so we're dealing with anything with designer babies has to be ruled out for the reasons that I brought up. You know, one of the things is that what we think is cool today may not be cool 10 years from now. And so a kid, a mom goes, I want to have a kid who's 6'5", because of all the advantages. But what happens if the kid wants to travel a lot? I mean, it's incredible. I'm getting on a plane in a few hours. You see these men, big, 6'5", and they're in an economy, and they can hardly get in there. And I have one of my brothers is 6'4", and he goes, he has to pay when he has to fly to Europe. He's basically just walking around the airplane while I'm passed out in the back. He's walking because it's so uncomfortable. So what you think is an advantage in one context is a disadvantage. Why? Because the world is constructed for the norm, for the average. As soon as you go beyond the average, the world is not easy to live in. So I'm just going to, these are just conclusions. For acknowledgement, this is the same acknowledgement slide. This is my lab. They want me home. They're texting me already. They want me to be home before they leave for Columbus Day weekend because we discuss some science. Wonderful, wonderful gift from God. And again, these are the grants that support my research, including a grant from the BioLogos Foundation, which we're working on mystic evolution. And I'm trying to find like-minded Catholic scientists who will help us in this project. So thank you very much for your kind attention.